Welcome back. This is question 6 of January 2025, Unit 4. The development of antibiotic resistance in bacteria is a global concern. A new type of drug is being developed against bacteria. This drug has a number of effects on bacteria, including making the bacteria more susceptible to antibiotic. The drug is an antibiotic. State the meaning of the term antibiotic. So, what is an antibiotic? An antibiotic is a chemical that is used to stop or inhibit the growth of a bacteria or a microorganism. Okay, so let's see the answer. An antibiotic is a chemical, substance, a drug, or a medicine that kills or inhibits the growth of bacteria, microorganisms, or pathogens. So, let's skip to the next question. In 2019, 1.27 million deaths were due to antibiotic resistance. That's concerning. It is estimated that this will rise to 10 million per year in 2050, which is the percentage increase in the number of deaths due to antibiotic resistance. So, the formula of calculating percentage of something is final minus initial divided by final into 100. Okay, so by using this formula we can figure out the percentage increase or decrease. Okay, so let's see. So the correct answer is C, 687%. Okay, now let's move on to the next question. Explain why it is important that these new types of drugs are developed. Why it is really important to develop these new types of drugs, so that bacteria does not become resistant to the new drug, and we will be able to treat antibiotic resistant bacteria or bacterial infections or diseases. If the new drug is not an antibiotic, then bacteria may not develop resistance to it, and it can increase the power of how the antibiotic works. Okay, so now let's see the answer. Bacteria will not be resistant to the new drug, and therefore we will be able to treat antibiotic resistant bacteria or bacterial infections and diseases. If the new drug is not an antibiotic, then bacteria may not develop resistance to it. Now let's move on to the next question. The diagram shows the structure of the cell wall of a bacterial cell. It also shows the cell membrane. The molecules in the outer membrane vary in different types of bacteria. So this is a bacterial cell. I mean the outer cell wall of a bacteria. And this is the outer membrane. This is peptidoglycan. This is cell membrane, and this is cytoplasm. Okay, inside of the cell. When bacteria were treated with the new drug together with antibiotics, the antibiotics were effective at lower doses. The new drug attached to the outer membrane suggests why the new drug resulted in the antibiotic being effective at the lower doses. So, what can be the answer? So the questions ask why the new drug resulted in the antibiotic being more effective at lower doses. The possible answer can be that the drug increased the permeability of the bacteria's membrane, so that's why more antibiotic can get inside to it, have more access to it, and that's why less antibiotic is needed, and the drug also act as a channel protein, we can say that, which opens the pores and more antibiotic can enter through it, and that's why we need lower doses of antibiotics. So let's see the answer. The drug dissipated or increased the permeability of the bacterial membrane, cell wall, or peptidoglycan layer. This allows antibiotic to access or affect its target sites like ribosomes, enzymes, cell walls, and enter the cell. As a result, less antibiotic may be needed to treat the infection. The drugs act as a channel protein, helping the antibiotic to enter. Okay. Now let's move on to the next question. The graph shows the effects of 3 antibiotics on the species richness of gut flora. So this is 3 types of antibiotics that affect on the species richness of gut flora. Okay, the bacteria which lives inside our intestine and compete with the foreign particles and eliminates them. Describe 2 conclusions that can be made about the effect of these 3 antibiotics on species richness of gut flora. So we just need to explain whatever we are seeing through the graph. Okay, so we can say that all the antibiotics, like all the 3 antibiotics, decreases the species richness. It shows a negative correlation. The effect of all 3 antibiotics is absolutely nearly same during the first 3 days. We can see it through the graph, and but we can also see a different trend when it comes after 3 days, that the antibiotic 3 causes greatest decrease in species. Okay, so now let's see the answer. Overall, all 3 antibiotics decreases species richness. The effect of all 3 antibiotics is nearly the same during the first 3 days, and antibiotic C causes the greatest decrease in species richness, destroy the most bacteria in the most effective and killing bacteria overall even after the 3 days. Okay, now let's move on to the next question. Explain the advantages of adding this new drug when treating a bacterial infection with antibiotic C. So what can be the advantages of adding that new drug that the scientists discovered with antibiotic C? We can need lower doses of antibiotic C. It will work faster due to as the drug works as a channel protein, and the species richness of the gut flora may decrease as much. So which means that the gut flora will continue to compete with the pathogens for space and the nutrients, aiding digestion and overall gut health, and as a result the patient will recover sooner from their infections. Okay, so now let's see the answer. And the antibiotic C may act faster and work at lower doses, and therefore the species richness of the gut flora will decrease as much, and which means that the gut flora can continue to compete with the pathogens for space and other things which aid in the digestion and overall gut health, and which will result in the patient being recovered soon. Okay, so I think this is the end of question 6. Now let's move on to question 7. Okay, bye.